Good afternoon, everybody. My name is Masarykova. I'm a senior scientist at the Institute of Vertebrate Biology at Czech Academy of Sciences. For those who don't know me, I would call myself an ecologist who is interested in primate parasite ecology. And within our the latter Czech-Japanese project, sponsored or supported by Czech Academy of Sciences and Japanese, what's the English?
Society for the Promotion of Science.
Japanese Society for the Promotion of the Science. We got a bilateral grant in order to improve the morphological analysis of the adult worms, and in order to transfer technologies, transfer knowledges between Czech and Japanese side. So the Czech researchers and students go to Japan to learn a classical morphology, and the Japanese students are coming to Czech Republic to learn molecular biology techniques. And within this project, we have invited several Japanese, although they don't look very Japanese, scientists to come to give a talks. And you may remember Mike Hoofman, who gave a talk in October. And today we've invited Andrew MacIntosh from the Primate Research Institute of the Kyoto University. Andrew is my long-term colleague. We work together on complexity of behavior. You may remember one PhD wiver, Jade Burgunder. She worked on complexity in behavior and parasites in chimps and sheep. And Andrew is going to speak mostly about the behavior part of our research. And I think it's enough, and Andrew will tell you more.
Okay. Thanks very much, Clara, and thank you all for being here. I hope we can go on a bit of a journey together for the next, how long are these supposed to be? Maybe an hour-ish? Okay. You can see how well prepared I am for this, so about that time frame. So thank you first, Clara, for the invitation, and it's really a pleasure for me to be back. I visited Brno about 6 years ago as part of a different project. So as Clara mentioned, we have been collaborating for quite some time, and hopefully we can push that going forward as well. So I'm going to talk a little bit about one branch of my research, but maybe not the main one. So I am based at Kyoto University's Primate Research Institute. So probably you pegged me as a primatologist because of that. I'm only partly a primatologist. I have much broader interest than that, but it's proved a nice base, which has allowed me a lot of freedom to explore different kinds of scientific ideas, using primates as models, but also other species. So primarily, I am a behavioral ecologist. So in this talk, that should be kind of the central theme for everything I'm going to talk about. Although, I also dabble with Clara, for example, in infectious disease ecology and epidemiology, and various subtopics like that. So my research has kind of taken me all over the world, and I just show these things just to get a general introduction of who I am and where I come from. So I've predominantly studied primates around the world on basically all three of the continents where they occur. But in this talk, you'll see me talking a lot about penguins as well. So it's kind of hard for me to sit here and find something that unifies primates and penguins, apart from the letter in the alphabet that starts both of their names. But hopefully, through this talk, you can kind of see how I got there. Okay? So I want to start this presentation by introducing you to a concept. So maybe many of you have heard of this concept before. It's not really a new concept. It's something that was really heavily in development in the late 80s, early 90s. But this idea that life itself, and life really being a representation of a complex system. So life itself exists at the edge of chaos. So as a student of biology and behavioral ecology, in my early days, I never really thought that my life would intersect with concepts from physics. But I was quite surprised when it happened, And so over the years, I've developed these kind of, I suppose you could call them role models or even idols in certain scientific fields. And so here's one of them, Stuart Kauffman, who's quite a famous physicist and also popularizer of concepts from physics applied to kind of everyday things. So he noted, this was in the 90s, that complex systems are generally constructed so that they exist at this kind of boundary between order and chaos. And so this is really the foundation for what I want to talk to you about today. I guess I can use this. So if you look at the video that's being played here, of course it's very beautiful. You can watch these kind of things for hours. I myself just took a video of something similar in Kyoto with Kyoto Tower in the background, so it makes for quite poignant visual representation here. But there's a couple of things that I want to highlight about this. So what you're looking at is a murmuration of starlings. And the kind of behavior that you see, of course birds are flying, we know birds fly, birds flock as well. But the kind of behavior you're seeing is a collective kind of behavior where you might describe the overall movement patterns as something that's very, very hard to measure. And that's true. It's very hard to measure in our traditional sense how the birds are moving. But what is very clear is that each individual bird is really only paying attention to a very simple set of external stimuli. So one of the main rules for how birds produce these kind of flocks is individuals basically tracking the movement of their neighbor and responding to the actions. And so because of those very simple rules, you end up having these very huge, high or large scale kind of chaotic looking emergent properties. So in today's talk, there's really two central themes that I want to keep in mind. So the first one is about behavioral organization. So a simple definition of behavioral organization might be the one presented here by Lucy Asher, where we can think of the arrangement of behavioral states relative to each other in time and space, and in relation to other individuals, locations, or behavior. So the bird flock is a good example of that. The birds pay attention to what other birds in the flock are doing, specifically their neighbors, and collectively they produce this very organized yet complex pattern of behavior. And secondly is that the modern way of describing or investigating these kinds of complex behavioral patterns is through what's known now as complexity science or nonlinear dynamics. And so this is where the physics really comes in. I am not a physicist, so I'm talking about a lot of physics concepts, but please do not grill me too much if you have a physics background on the specific details. But anyway, so complexity science gives us a new way to study these complex regularities in nature. And it is thought that this is kind of one of the frontiers now of behavioral ecology to really help us understand, not just simple patterns of behavior we observe, but how those simple patterns scale up to the very complex patterns we see all around us. But ultimately, we see that a lot of the complexity in the natural world really does start with very simple rules. And so in today's talk, I want to use some of these concepts and walk you through a little bit of chaos and fractals in animal behavior. So probably you've all heard the term fractal and chaos. This is kind of a simple cartoon way of illustrating what fractal is, but basically you have these kind of self-repeating elements in some kind of system, either a structure or a process, that doesn't matter really what scale you're looking at it, it tends to look the same across all of those different scales. And hopefully you'll see that by thinking about this concept, we can actually unify or find a unifying principle that can help us describe what behavior actually is and how it works in structure and in time. So fractal geometry was really started maybe in the very late 19th and early 20th century, but it wasn't really all put together until the 1960s when a famous kind of the father of fractal geometry, Benoit Mandelbrot, who I'll introduce in a second, started to put these ideas together. But basically, the reason this is useful for us is if you look at all of these are what we call mathematical fractals. ideal fractals. So there are very specific rules to generate all of these very complex looking processes. But if you think about this line, so this is the Cantor set. You can take a solid black line and you apply a rule to it, an algorithm that at each step you divide the line into three, or you remove the middle third, of the line. What you see is this thing becomes very hard to measure because as long as you have a line, you can always divide a line by taking out the middle part of it, middle third of it. So that means it's kind of an infinite process, which at the time in mathematics really confused a lot of people because, of course, infinite things are not measurable. But that's kind of what set the stage for this kind of mathematics. And then I have some other examples listed here. But what's really interesting is that, if you think about it, a lot of the things we see all around us in nature, so they end up being natural fractals. So the kind of sky, if you look at the sky, and the clouds in the sky, they kind of at the beginning defy measurement. Same thing with like coastlines or mountain ranges or cliffs. The patterns tend to defy traditional measurement, and I'll explain that in a minute. And same thing for the branching patterns of trees. We know that the processes that arise or that cause these things to emerge are not super complicated, but yet the resulting patterns are very complicated to measure. And so that's kind of the background for what I want to look at. So an example of a fractal in action here, so this is the Mandelbrot set, and it kind of illustrates the process. So at every time you try and kind of get higher and higher resolution, we always end up with the same patterns repeating themselves over and over again. This is the same kind of process that kind of generates these natural structures you see here. And by the same token, if we start with a triangle, we can have a very simple algorithm that, if repeated ad infinitum, ends up with something very complex, like a mountain range or something. And so we can use these concepts to try and understand how different patterns might be generated. So a second ago, I introduced this gentleman, Benoit Mandelbrot, who, as I mentioned earlier, I didn't think I would have too many idols in physics and mathematics, but it turns out that this gentleman was one of them. And he famously quoted in one of his books that clouds are not spheres, mountains are not cones, coastlines are not circles, and bark is not smooth. So basically, nature exhibits not simply a higher degree, but an altogether different level of complexity. And the complexity in nature tended to defy a lot of measurements of it in the past, because we can't impose kind of more traditional Euclidean geometry onto the structures we see, or things like that. And so this gave us, Mandelbrot started to give us new ideas and new tools for being able to measure natural structures and processes. And so here's an example of how he did that. He asked this very simple, seemingly very simple question in the 1960s, which was, how long is the coast of Britain? So I'm not sure that's something that many of us in this room would have considered or consider today, but it's actually a very philosophical question. And the reason is that the length of anything really, especially when it's something as complicated as a coastline, really depends on your measuring stick. And so basically what Mandelbrot was able to show is, if you use measuring sticks here of, let's say that's 100 kilometers or something, probably not, but anyways, the measurement for the length of the coast that we get is going to be smaller than it is if we use 50-kilometer measuring sticks. And that's going to be even longer again when we use 25-kilometer measuring sticks. So the point is here, depending on the unit of measurement that you're using, your end result for the length of the coast of Britain changes a lot. And so what he suggested then is the absolute length is not really a valuable measure, because you can kind of create any length that you want depending on the measurement scale that you have. And while he was doing that, he also showed how, if instead of looking at the absolute length, let's look at how the length changes as a function of the measuring stick. And when he did that, he noticed that there was a scaling factor involved. And so basically the measurement scales here, as this is a fractal dimension estimate that he concocted. So the measurement changes as a factor of 1.2 fractal dimensions, basically, as you change the scale. And actually, that turns out to be very simple to one of those mathematical fractals, the Koch snowflake. So what he basically did was he unified measurement terms in fractal geometry and pure mathematics with an ability to measure natural structures like coastlines in the real world. And so since then, there's been kind of a lot of interest in describing these scaling relationships, these fractal dimensions, in a lot of different kind of systems. So you can just see a table here, which you don't have to pay too much attention to, but ultimately you go from coastlines to galaxies, to trees, waves, clouds. Over here you have some physiological structures like retinal blood vessels, even things like bacterial growth patterns, electrical discharges, all kinds of different things which were rather complex and hard to measure in the past could now be described by these scaling properties that really talk about how their structures are kind of unified across different scales of measurement. And so by doing so, we can, instead of looking at some certain absolute properties which might not exist, we can look at those scaling estimates and compare those across different systems. So let me kind of exemplify this in a different way. So one of the famous applications of this fractal dimension estimation came out in the late 1990s, early 2000s, when there was so this is a Jackson Pollock image. It's called, the painting is called Number 14. It's quite a famous painting. But this idea of using physics to measure some properties of natural things came really to the forefront of our society when there was a new painting or a new cache of paintings discovered which were supposed to be attributed to Jackson Pollock. This was long after his death, and of course, if a painting set has the name Pollock attached to it, the dollar value increases on the order of millions or tens of millions of dollars. And of course, if it's not really Pollock, then somebody can just do what they will with them. And so this physicist, Richard Taylor, said, you know what, why don't I take a look at them, and I bet I can tell you, or I find a way to tell you whether these are actually Pollock paintings or not. And so what he did is he looked at the evolution, the time evolution of Jackson Pollock paintings, and he measured the fractal dimensions, so basically the complexity of the image as a function of scale. And he found that as Pollock aged, the kind of complexity of his paintings were kind of increasing over time. And so the time when they found the cache of paintings from Pollock, or the alleged cache of paintings, this physicist, Richard Taylor, looked at those paintings and did the fractal dimension estimate and found they actually fit exactly on the curve, which was predicted by his own kind of evolution of painting. And so by using those techniques, he was able to say, these are probably Pollock's. Whether the art community accepted that unconditionally, I'm not sure. But it's an interesting way to think about measuring works of art. But kind of more importantly for our purposes in this talk, and this has also come out with from original studies in physics that were looking at complex systems dynamics, is this question right here. So if certain physical or complex systems have these measurable dynamics, can we use them to ask important questions about systemic health? Okay? And so the question that arose from those studies is there a significant change in the fractal dimension when measured for systems operating with certain malfunctions? Okay? And that becomes really important for what I want to talk about here. So again, in the 80s and 90s, some physiologists and human health practitioners started getting on this idea as well. And so they were looking at different physiological structures. So here you have three. You have the vascular system here in the heart, you have the lung architecture, and the structure of a neuron. And in all of the cases, they produce these complex kind of fractal-looking structures where the patterns kind of repeat themselves at different, increasingly small scales. And what they found out with those is that the physiological fractals here exhibited altered dimensions this is cut off here, become pathological. The systems become pathological, they exhibit altered dynamics in their structure. And so one of the most famous examples of this to the point where some physiologists were trying to push this to becoming a diagnostic tool for human patients with certain heart conditions. But basically, if you look at these three graphs here, these are just the interbeat interval patterns or heart rates for a human subject. And so what you can see over time here, this is 300 minutes of a heartbeat sequence. This is 30 minutes of a heartbeat sequence. This is 3 minutes of a heartbeat sequence. And basically all three figures look the same, but they're taken at very different scales. So that's kind of the essence of what a fractal is. You know, you can change the scales on order of magnitude, but the pattern looks the same. But what the scientists ended up finding was, if you look at these interbeat intervals for healthy human hearts, so here we have three columns in these figures. They represent a normal, healthy heart, heart in a pathological condition, kind of early stages of some pathology, and then a heartbeat in the later stages of pathology, just before a cardiac event. So the pattern in the beginning looks pretty complex, actually. You have a lot of variability in the beat-to-beat intervals, and sometimes you have spikes. In the early onset pathology, you see it starts to dampen a little bit, so that the heart rates become much more periodic. And then eventually, they look like what we call a damped pendulum, so just the swinging of a pendulum slowly going to zero. And so the bottom graphs here are what we call strange attractors. So in physical terms here, this healthy heartbeat creates what we call a strange attractor in phase space. So just a really complex pattern. There are some repetitions, but they're not constant. But as you go towards pathology, you get less and less variability in the heart rate. And so this is why these physiologists wanted to use this concept to help us understand or diagnose hearts operating in poor conditions. And ultimately what they found was, going back to Mandelbrot's terminology, the fractal dimension of a healthy heart is greater than the fractal dimension of a diseased heart. So in principle then, you can use the fractal dimension, which is this index of complexity, as a measure of kind of viability in a system. And so since then, there's been a lot of interest in kind of broadening the utility of this kind of approach to different kinds of systems. And there was a really interesting paper published 12 years ago now in Nature by some Spanish colleagues who were kind of interested in this idea of, can we use the same kind of complexity indices to look at patterns of change in vegetation structure. So one of the current, very modern issues of climate change especially, is the conversion of certain landscapes to desert landscapes, right? So this is a process called desertification, and it's happening, so this is in a few different regions in the Mediterranean where they did these studies. Basically, they look at when you have kind of quote-unquote healthy or optimal looking vegetation patches, vegetation structure, the distribution of the size of the patch of vegetation and its frequency creates a power law. So here's the frequency of occurrence of those different patch sizes, and this is the size of the patches. And you can see that in a healthy kind of ecosystem, you have a power law relationship between those two things. But as you go further and further towards desertification, so much less vegetation structure in the landscape, and mostly this is caused in the Mediterranean region by overgrazing. And so that was the question, is what influence does overgrazing have on the vegetation characteristics? But you see that the actual distribution here, this relationship between size of patches and number of patches, changes from a power law to a more kind of, they call truncated power law, or even getting close to an exponential distribution. So again, basically what you have here is a significant change in as well the fractal dynamics or statistics in the vegetation patch structure as a result of this stress in the system caused by overgrazing. So these are just a couple of the past applications of this idea to help us find a way, if we can predict, if we can use those concepts to make predictions about a system which is on the verge of some kind of collapse. Okay, that's the central theme for the talk that I'm giving today. Now, when it comes to animal behavior, it has been noted in the past that behavior should be viewed, or could be viewed, as a complex adaptive system. So it's a system that engages, or that allows an animal to engage in different ways with its environment, responding to its environment in different ways. And what we know from the earliest thinkers in the study of animal behavior, here you have Konrad Lorenz, of course, who wrote in his famous book, Foundations of Ethology, the intensity of so much instinctive behavior. And you remember those fathers of ethology were very much interested in instinctive behaviors of animals. It really depended on the physical condition of the animal. So he's talking about when we take animals out of their natural environments and we put them into different captive scenarios, we all know that the behavior changes drastically and sometimes rapidly. So it's hard to use the animals in those environmental conditions to represent individuals in the wild. But I think we can go farther now that we've come a long way since these times and argue that not only the physical condition, but also physiological condition, psychological condition, in combination with biotic and abiotic components of the natural environments, all have a big influence on the patterns of behavior that we observe animals to produce. And so this, for me, is kind of the entry point into my research, which is really focused on how we can use those concepts derived from statistical physics and apply them to monitoring animal behavior in real and artificial conditions. And so that's where this idea of trying to look for complexity loss, so these features in the dynamic processes of behavior that I've been talking about, like the heart rates or the vegetation structure, can we find those same kind of signatures in sequences of animal behavior when they're operating in certain kinds of pathological conditions? And so not just our research, but a host of other research has really shown over the last couple of decades that yes, we can. So in certain toxicological settings, in relation to stress in the environment, in relation to physical handicaps or even human disturbance, in relation to natural influencers of behavior like health, pregnancy, and parasitism, across the board, we've started to see these altered fractal dynamics in behavior patterns, almost wherever we've looked for them. And what's really interesting for me is that we can really break down behavior into simple sequences of numbers and then apply the same kind of unifying concept to find the same results, even when the systems are very different. So that's, I kind of want to get a little bit into that as well. But the reason that I initially got interested in this kind of research was because for any of you who've actually gone out and observed the behavior of animals in nature, it's not always clear to us which components of behavior actually might indicate some kind of a stress on the system or some kind of a condition indicator, which is problematic because we often like to use the behavior of other species, including human behavior, as what we call a bioindicator, right? And so, or an eco-indicator of some kind of environmental issue or ecological problem. So bioindicator is any species, which is called an indicator species, or group of species whose function, population, status can reveal the qualitative states of some environment. I would argue that behavior is also one of those things. We can use behavior to indicate the quality of the environment. And so a biological monitor, or a biomonitor, is an organism that provides some quantitative information about the environment that it's in. So as I mentioned earlier, if we think of behavior as a kind of complex adaptive system, we can really see how behavior itself is that bridge, right, between an animal, its environment, and its environment. And so it's this concept that I want to elaborate here. But as I mentioned, when we're stuck, or when we're faced with looking at animal behavior, especially in natural conditions, there are so many different kinds of things we can pay attention to. It's not always clear to us which of those things are going to give us the right kind of information that we need. So if we just take penguins for an example, and I'll give a lot more from penguins a bit later, we can measure things in the natural behavior of penguins, such as foraging efficiency, foraging effort, dive duration, trip length, number of dives, dive depths, trip distance. The more I've been involved with research on penguins, the more I've seen the range of different kind of things we can measure. And of course, if you take any animal, you can imagine the same breadth of topics that you might be interested in looking at. But it's hard for us to kind of pick and choose which of these kind of behavioral measures would be the most relevant to us, especially when we want to use those animals as a biomonitor or an eco-indicator. And so, I don't know if any of you are familiar with a couple of popular media here, but we've got the issue of trying to decode the matrix into some value that actually makes sense to us. And if there's any Douglas Adams fans in the audience, you might know what I'm talking about here. So, enter some relatively recent research starting from about 15 years ago, really. More and more people have started to recognize that kind of by using some of the statistical physics measures that I've already talked about a little bit in some different ones, a lot of animals are producing the kinds of behavior patterns that we can simplify. And when we do so, using kind of common methods from physics, like looking at distributions, for example, of behavioral outputs, it turns out that a lot of different things look the same. So a lot of different species and a lot of different environments tend to produce behavioral outputs or emergent properties of behavior that look pretty similar. And so there was this famous paper in 2007 that came out that talked about the scaling laws of marine predators' search behavior. And that was linked to this kind of emerging idea that statistical patterns in animal behavior suggest that more complex, or we call them Levy-like or power law distributed behavior patterns, might look like an optimal search strategy. So the idea, it looks like this, mostly by using technology to measure animal behavior in situ, right? For animals we traditionally don't have ways of accessing. So here's a sea turtle with kind of GPS and accelerometer glued onto its shell. But over time, if we're measuring that, we can get these really high-resolution behavioral sequences that give us a time series here. This is the movement step length, so how far the turtles are moving within some given step distribution. And when they map those out, you end up with this kind of relationship here. So this is the size of the movements, and this is their frequency. You can see this looks pretty much like a power law distribution. So most of the movements are really short. A few of them are really long. But the relationship that describes the frequency on the log scale of those movements to their step lengths ends up being like a power law here. And so this study found that from species as diverse as sharks, turtles, and penguins, basically those movement properties all look more or less the same, right, in very simple terms. And we can use the slope of these log-log relationships, these power law relationships, which might look something like this, as a way of describing their behavior and comparing between different species and conditions. If we think of the Jackson Pollock painting here, number 14, it had a fractal dimension of something like 1.3. But at the time, for animal movements at least, there was a real interest in determining whether we can describe animal movement as just a result of Brownian motion. So are they just following random rules to define their search, or does it look much more complex and power law distributed like the Levy walk here? And for the most part, we tend to see this pattern is more dominant to the Brownian pattern in nature. So here's an example of kind of a follow-up study that was really testing whether these different kinds of movements, so Brownian movements versus power law Levy-like movements, how do they relate to different kind of environments that the animals are in? And so here you can see a few examples. So this is black-browed albatross data collected, again, using biologgers at 3 different scales. And at each of these different scales, so here you have the section blown up here, and then this section blown up here. The movement tracks themselves look pretty similar across all of those. So the patterns are, in a sense, fractal. right? They repeat at different scales of measurement. And more than that, the study noticed that the albatross were actually switching between kind of localized random search when they found maybe a resource patch, and levy-like kind of broad-scale search when the environment was a little more heterogeneous. And so this prompted the authors to argue that in heterogeneous environments, so that's where prey might be further between and kind of more patchy in distribution, having a more levy-like or power law search strategy could be quite useful. It was since really challenged heavily by biologists, saying that it doesn't really make sense to have this kind of power law generator in the biology of the animal. So recently, just a couple of months ago, the same group of authors published this study, which was looking at fruit fly larvae, basically. And they were looking at the movement of these fruit fly larvae here at really high resolution. So looking at their turning angles and the paths that they create. And again, here's the example of a fractal. So this is one scale, a smaller scale, a smaller scale. It all looks more or less the same. But what they discovered was that by blocking activity of the brain here in the central nervous system, the larvae are still generating power law movement distribution. So even without a brain. So that means they're not responding to the environmental cues at all. They argue that this is like a central kind of pattern generator, which must have been evolved in the biology of the organism to create these really complex movement strategies. So anyway, this debate trudges on, but it really adds, I think, to this two kind of main key concepts in kind of fractal dynamics in behavior sequences. So the first one, which I've just discussed, is that it seems like fractal patterns might be optimizing search strategies. And the second one is that, as I started with, error tolerance might be a really important part of having these fractal behavior sequences emerge, because it's the most error tolerant systems that are most able to adapt to changing internal and external conditions. Okay, so these are kind of the two hypotheses behind the emergence of fractal patterns in behavior. Okay, so a few vignettes from our own work, after that rather long and lengthy introduction, I'll get into now. So basically the models that you'll see come up here are a couple of different species of penguins, some primates, and the domestic sheep over here. And so I want to walk you through how our studies reflect these emergent fractal patterns in behavior in relation to the role of the environment in evolutionary constraints and the role of individual conditioning. Okay. So the first thing I want to walk you through is a little bit of methods. So basically how we generate these complexity, I'll call them complexity profiles, is basically what I mean is the emergent kind of fractal statistics that we can pull from behavior sequences. So generating these, we've been doing them in a couple of different ways. So with species that we can't traditionally follow in nature, such as seabirds, penguins as an example, we're exclusively using animal-attached devices. So these range from GPS loggers, accelerometers. A lot of my studies are really based on time depth recorders, where we can monitor the depth of the animal at any given time step and look at the profile of dives that way, and then turn them into a time series, basically. And so biologging allows us to get very up close and personal with the animals in their natural environments and extract a lot of high-resolution data from them. And so the two model systems I've been using are little penguins in Australia and the Adelie penguins in Antarctica. And so if you just go back a second to the levy-like foraging hypotheses studies, basically they're doing similar things. So with animal-attached devices, looking at the sequences of behavior here, this one's flight speeds in meters per second. So you can have an indication of when the animal is flying and when they're not. And so that gives you this continuous series, which could span weeks to months of behavioral data. The other way we're doing it is in very traditional terms here, especially with our primate studies. It's not always so easy to attach devices to primates for various reasons. So we just get into the forest and watch them ourselves and record behavior continuously. And when we want to think about how the data are analyzed, I'm just going to introduce you to one of the many methods that could be used in fractal statistics. And I'll give the example with penguins. So if we have a sequence of behavior, and maybe what I'm interested in is whether the penguin is diving or not diving, and sitting at the surface. So we can generate a sequence over time. The data structure might look something like this if we binarize it. It's a very simple kind of, and penguins lend themselves well to this because they are a binary system in, when they're foraging. They're either under the water or they're not. Of course, that's a bit facetious, but anyways. We end up with this binary time series, and if we integrate that time series where we accumulate, the dives versus not dives over time, we end up with something that looks like this. And then ultimately, all we want to do is measure the fluctuation of this dive sequence over different scales. So we can keep increasing our window of measurement, we keep taking a slope of the fluctuation within those things, and then we can compare the average fluctuation as a function of scale, and we end up with something that looks like this. And traditionally, or typically, it ends up looking like a power law. So we can use the power law exponent, or the scaling exponent, as an indicator of the complexity in that sequence. And what I have been most interested over this time is figuring out, is there kind of a Goldilocks zone? I'll get into this a bit later, in complexity within sequences of behavior. Is there a point where there's too little or too much complexity, and what does that actually mean? So if you think of this, this could be a range of complexity, of scaling exponents here. This 0.5 represents completely random behavior. So if you think about how rough that behavior is, total randomness is very rough. So that's what this bell-shaped curve is indicating. But in terms of complexity, randomness is not really complex. But neither is complete determination or determinism. So we expect behavior of most complex systems to fall within a range, either anti-persistent, where sequences are, over time, they're less like what happened just before, or what we call persistent, where they're more like, so positively autocorrelated through time. So this is kind of what gives us a way to interpret what those scaling exponents or complexity signatures mean. And so in the real world, if we look at penguin diving behavior, we can create sequences that look like this. This is a real penguin dive sequence in binary time. So the black bars indicate diving, the white bars indicate no diving. And if we add those sequences all together, here's 5 different penguin foraging sequences, you can see that there's a lot of fluctuation within individual sequences, but also between individuals. So these 5 different individuals are creating fractal profiles that look quite different from each other. And it's this kind of variation that allows us to test the factors that can predict different types of sequences we see. So one example I can give you about environmental influences on this is we've looked at little penguins, so it's the same species basically across its range in Australia and New Zealand. And 4 different colonies are represented here, and the kind of bathymetric features are illustrated up here as well, where you can see that the Penguin Island with the contour map here is a little bit soft in terms of the topography under the ocean, or the bathymetry, whereas Phillip Island is a little bit more heterogeneous here. And that becomes important apparently when we're looking at the complexity signatures of the different birds. So we found that here, Phillip Island in Australia, just near Melbourne, the birds ended up producing kind of the most stochastic sequences of behavior. So these are the least long-range dependent, a little bit more complex in terms of the predictability of the sequence. But it also turns out that that is the colony that has the lowest breeding success of all of those 4 in the year that we measured this. And so that's some indication maybe that we can link the foraging sequences with breeding success through these kind of fractal patterns. And what we've learned over a lot of our analyses is that for little penguins as well, if we use measures like more traditional measures, so catch per unit time is an estimation of how successful the foraging sequence has been for a little penguin, or the cumulative foraging effort, which is the amount of time they spend underwater, we tend to see here positive relationships between this fractal index. So more deterministic sequences are associated with higher catch per unit time. So more deterministic sequences seem to be more successful. Whether that's causal or a result is something we don't know yet. But it's quite contrary to the case for foraging effort. So it looks like with increases in foraging effort, it's associated with kind of increases in the stochasticity of behavioral sequences as well. So that might be indication that producing more random sequences has a cost. Right? And so that's something we kind of continue to explore. But one thing we also are very much interested in is not just the different environmental conditions, but what about the evolutionary constraints on the bird? So penguins, there are about 19 species represented right now. And so we've put together a data set that looks at, these boxes are a bit off, that looks at 11 or 12 of these different species. And we're trying to understand how much does the phylogeny, so the shared ancestry, predict this variation in scaling across birds. And so we've created a data set here from penguins pretty much across the Southern Ocean here. And a lot of co-authors have contributed data to this. You can see we're over 100 million data points, which is pretty interesting. And we found that there is a really high phylogenetic signal. So a lot of the variation in behavior patterns and scaling that we see can be attributed to the phylogeny, so shared ancestry. But if we remove that phylogenetic component to the analysis, the things that mostly drive variation in complexity are whether the penguins forage for active or passive prey. So if they're foraging for active prey like squid or fish, the complexity signatures tend to get higher, right? A little more randomization in there. Lower latitudes as well, and in smaller size. So the smaller penguins tend to also have more stochastic behavior patterns. But phylogeny seems to be the main determinant of this across species. So that was pretty interesting to us. So it does seem like, if we think about variation in complexity across species, there looks like there might be some kind of Goldilocks zone. And this term comes from astronomy, like looking for habitable planets, basically. They tend to be situated at a certain distance from the sun of any solar system, like our Earth. And so there looks to be this habitable zone there. It looks to be this kind of Goldilocks or optimal zone for complexity in penguin sequences. And so when we've looked at this in terms of relation to breeding success, so we've done a study where across a decade of research, or more than a decade, 15 years of research with little Adelie penguins in Antarctica, it seems like the periods with kind of moderate sea ice concentrations, Adelie penguins are heavily dependent on sea ice for their breeding success. And you can see lower breeding successes with less sea ice and also with greater sea ice. So that Goldilocks zone for breeding seems to be in this sea ice concentration range. We've also seen in terms of the complexity signatures that there seems to be a leveling off point as well. So we expect that there might be some evidence for these Goldilocks zones in behavior as well. But behavior is much more variable across time, even within seasons, than something like breeding success is. So it's not clear to us exactly how important the behavioral profiles are for predicting breeding success in total. So this is kind of what brings me to this idea of complexity loss and what we can learn from it. So if Goldilocks zones in these fractal sequences exist, then what is it that sets animals off or away from these kind of optimal zones in their behavior? And then what can we learn from that? So I'll just give a few more vignettes about studies of complexity loss here in our own work, looking at logger attachment, corticosterone implantation, reproductive energetics, and then ill health and parasitism. So like a lot of modern studies that have been using animal-attached devices, the first thing for us was to really look at, with penguins, how much do the devices themselves actually influence the behavior we get? And it turns out that it's not negligible, right? So especially with smaller species where the size of the logger would have a greater impact on a small penguin, which is less than a kilogram, than a rather medium-sized to large penguin at 3 to 5 kilograms. So you can see that larger loggers ended up creating these altered fractal dynamics in the behavior sequences, it basically introduced behavioral noise. So the birds were more stochastic when they had these loggers on them. So it's kind of an important thing to recognize when we want to make comparisons across different systems. But it also gives us a proof of concept. If we introduce some kind of perturbation, these are the kind of outcomes we can expect. We've also done studies with Adelis in Antarctica where researchers were interested in the effect of stress on parenting behavior and on foraging efficiency and things like that. And so we used those studies, we leveraged those studies to do this complexity comparison as well. And what you can see in this chart with Adelie penguins is that for those control birds, this is, when we attach loggers, you can measure behavior for a certain number of diving trips or foraging trips. So we measured 4 different trips of each individual bird. And you can see that for control birds, there wasn't a whole lot of variation across the 4 different trips. But for the and these are birds that had corticosterone implants under their skin, which is supposed to be a timed release, which would release the steroid at a constant rate. I'm not sure how constant it was actually. But you can see that it induced a big change in the complexity signature of the birds. So that was measurable for us, and it's kind of an indicator that increased stress might have an important influence on those sequences of behavior as well. So here, they actually become more deterministic rather than more random. In my own kind of data generation work with primates, this is a Japanese macaque, a wild Japanese macaque. This one's on one of the southern islands. This is another proof of concept. We've learned that if you think about reproductive energetics, so animals for females that are reproductively active, there's obviously a huge energetic cost. So I was interested in whether that also translated into variations in the behavior sequences we can see as well. And so they're a seasonal breeder. So in the fall is the mating season, and then the spring is the birth season. So in the fall, the females are going through cycles of estrus. And you can see that the females that are exhibiting signs of estrus have kind of different complexity signatures here. A little less complex in this case. In the winter, that's when the females are pregnant. So again, there's a difference with females that are not pregnant. In the spring, when they have babies, again with infants, less complex. And then lastly, this is during lactation in the summer and carrying infants. So each different stage of the reproductive cycle seems to generate the same kind of change in the complexity of those sequences. So again, that's more proof of concept that we can actually measure measurable changes. We have measurable changes in fractal dynamics. And so we applied this also to animals in normal versus impaired health. So that was animals exhibiting clinical signs of poor health, like vomiting, lethargy, slow movement, that kind of thing. And we see very clear differences. So this is a monkey that was sick on one day and then kind of healthy the next. And her behavior changed a lot from day to day. If you compare that with other individuals on those 2 different days, we see most of the time it looks more like this. And what it looks like here is that the behavior of those kind of healthy monkeys had a different kind of complexity signature than those others. And so Clara mentioned that there was a graduate student here at Masarykova University that was interested in this as well, and she found that with chimpanzees in the wild, individuals that had higher intensity of infection with a certain strong-gall nematode, so probably a species that's very common in great apes, but with intense infections might create some kind of a problem. You can see that there is a subtle but measurable change in the scaling exponents of chimpanzees in those different conditions, and kind of demonstrated more strongly this concept with domestic sheep, where we used a deworming experiment where you had kind of baseline sheep in pre and post treatment periods, and that's the control line here. So there was a kind of environmental shift in the complexity signature of behavior, getting a little bit more, in this case, it's actually more predictable in the post-treatment phase. But for the treatment group where we dewormed them, it's massively different from the control sheep. So there was a big influence, apparently, of the parasites in the gastrointestinal tract maybe on the energetics. We don't know the mechanism, but a big influence on their behavior. So basically what I hope I've been able to show through this presentation is that we can say that animal behavior occurs in fractal time. We can use fractal statistics to measure sequences of behavior. And so it means that behavior occurs along this stochastic deterministic gradient. And we can measure how those properties change with respect to environmental and kind of condition dependence across individuals. It's still not easy to predict the direction of change that we observe, so that's something we're working on with more and more data coming in, or the mechanisms underlying it. But ultimately, the goal of this is less to understand so much the mechanisms underlying them, but more to be able to use these tools as a way to monitor and diagnose certain conditions. Right? So kind of my research program is really about this, whether we're looking at penguins or sheep or primates. We want to use these behavioral sequences we can generate in space and in time and be able to characterize the complexity of those sequences in a way that lets us understand animal responses to these different ecological challenges that they face, such as global change, climate change, habitat disturbance, even bringing animals into captivity in terms of measuring health and welfare, and looking at natural challenges like seasonality, disease, and competition. So if we have a range of information that gives us a way to measure these baselines, these Goldilocks zones in behavior, then it looks like we can use the complexity profiles in a diagnostic or eco-indicating way. And I just want to leave you with this last, hopefully kind of poignant example. When we've taken animals out of their natural conditions and put them into very artificial conditions, one of the outcomes that we often see if you go to the zoo is something like this. So here you have a stereotypical polar bear. Polar bears in nature should not be behaving in this way, although we'll see what happens when all the ice is gone. But this is clear evidence that we've already passed a critical threshold in that animal's life. We've passed the point where we can make the environmental conditions okay to elicit naturalistic behavior from that animal. And if you think about it, the example I gave with the human heart, this time series looks very much to the one being generated right here for this kind of pendular stereotypical bear. So one of the goals that I have, not just looking at animals in the wild as eco-indicators or monitors, but also can we use these kinds of statistical measurements of behavior to pinpoint times where it's still possible for us to change the conditions in a way that's beneficial to the animal and predict the onset of these massive phase transitions into what we can clearly see as complete catastrophic outcome. And so with that, I'd like to thank everybody here, thank the people that invited me, thank Masarykova University, the Institute of Vertebrate Biology, the Veterinary and Pharmaceutical University of Brno, and all of you for sticking with me. Thank you very much.
So any questions? Okay.
So we showed that acute stress response in penguins can increase this fractal complexity.
Yeah.
And how is it with the complexity in case of chronic stress?
Yeah, chronic stress, it's a great one. I mean, if we go back to the polar bear example, right, that I just showed, you can see the sequence that was being generated here. There's not very much variability in that sequence at all. If I take the measurement, it's actually a little bit closer to random. And so a random sequence, in some sense, does represent a complex sequence because it's not so predictable from one step to the next which value you'll see in the future. But at the same time, it's also very deterministic. So there's something very simple that's controlling the behavior so that it doesn't have any variability. Right? And so I think in this example, the bear clearly has suffered from chronic stress. So that might be one example. These stereotypical, really stereotypical behavior patterns might emerge. But I think we still don't have very good evidence yet for animals in chronic stress, so that's something we hope to start generating as well. I imagine, with the acute stress in the penguins, we see a transient change. So in the very beginning, there was a big effect on the behavior, but after that, it kind of leveled out. So maybe either the implantation wasn't so effective over time, or the bird compensated. That's normal in natural conditions, but who knows in others.
Thank you for a very interesting lecture. And I just wanted to ask, because you gave a number of interesting examples, it was almost immediately there are a number of human examples from psychiatrical disorders that spring to mind. Do you know about any systematic surveys in that area?
Yeah, so I've seen some studies with schizophrenia, actually, and they've used kind of similar analyses, not necessarily fractal, but time series analyses of, I forget the context, but it might have been choice patterns. And we've actually tried to replicate this, but the human psychiatric studies have looked at that, and you see with certain, like schizophrenia, for example, you do see very different patterns of those kind of correlation dynamics, or they use entropy, so the extent to which behavioral correlations degrade over time. You see them maybe happening more quickly with schizophrenic patients. We've kind of tried to replicate that with great apes. So I have colleagues who do cognition of great apes, and we're trying to put them in competitive situations where they're trying to match and mismatch choices against a competitor. And what we see is, depending on the algorithm we use to generate the correct choice, we can change complexity is the wrong word, but we can change the correlation structure of the choice patterns by great apes as well. So kind of mimicking those human studies. But I think that this kind of approach has been used in psychological work as well. So some of the people, I think in France, have been leading this idea as well. So, and then it goes from the behavior right back down to the neurological inputs. So a lot of neuronal firing has clearly been shown to have fractal properties, and that breaks down in pathological states. So I think there's probably a connection there as well.
Yes, and also for example, when you mentioned the schizophrenics, they often exhibit this flattened behavioral patterns that remind me of this swinging pendulum.
Yeah, exactly.
More like repetitive, deterministic.
Right.
But in other parts, it's more random and disorganized.
Yeah, it's interesting.
It's not a clear cut.
I mean, there's studies with human gait dynamics as well in Parkinsonianism, where they'd show that the patterns of movement become much more erratic using these kind of methods as well. So it does seem to have pretty broad application.
Or I think autism, because autistic kids are showing this stereotypic behavior also, engaged in certain, in this really same movements.
Yeah. And I think eye tracking, there's been a few eye tracking studies. I'm not sure with autistic kids, but that show very, very different kind of dynamics of the eye saccades as well.
So when you study with the small penguins, if I remember well, you concluded that actually it was the effect of body size, that the smaller penguins showed more stochasticity in the behavior.
That across the board, that seems to be the case.
So what's the interpretation? I mean, why couldn't it just because of the tracking device, or the smaller body actually can generate more stochastic movements or unpredictable movements?
My guess is that and it comes down to phylogeny, which is based on, adaptations to certain conditions. So smaller sized penguins tend to have diets that include more active prey. So I think there's a lot that we cannot separate, which is why I focus on the phylogeny. So if you look at the largest penguins, so emperor penguins, king penguins, a lot of especially emperors, a lot of the diet is krill. And krill is something that has these huge prey fields, right? That once they're in a patch of krill, there's not so much stochasticity necessary in order to manage that. So I think that's probably part of it. But the other thing is that small-bodied penguins might be more influenced by currents as well. So I don't know.
So it's the response to the external stimuli, either to the presence or behavior of the prey or the environment?
It's hard to say now because of phylogenetic constraints in evolution. So probably it's very much related to that. You know, animals being primed for certain types of prey.
About these flight patterns in albatross, which you showed. Well, I have to take your word for that it was fractal, you know, because it's maybe
Sure.
difficult to see.
Sure.
But what would be the adaptive use of that?
Yeah.
Okay, an albatross probably doesn't use that much energy while flying over the sea, so maybe it's not so important for this species than for others. But still, I mean, it's time. It should be feeding or doing something.
Yeah.
So, is there any reason why it should be fractal?
The hypothesis for that, and I'm not saying that I'm a proponent or antagonist to the hypothesis, but the hypothesis that I was presenting, this levee flight foraging hypothesis, suggests that the reason we see those patterns emerge is because they're just better at maximizing encounters with, for example, prey in heterogeneous landscapes. So if you have, I mentioned krill as one example. If you have a massive prey field and an animal is in the center of that prey field, then random Brownian motion is probably just as efficient as any other pattern to capture to maximize the efficiency or encounters with prey. But if you have prey fields that, you know, are heterogeneous in space and in size, then having an algorithm inbuilt through evolution that actually dictates animals, like not just making short-range movements within a patch, but making sudden long-range movements between patches. That is where the hypothesis is built on, that idea. So it allows animals to explore more efficiently. But that's been heavily criticized. So many of the criticisms are that animals responding to very simple cues in the environment also produce fractal dynamics. It's not that they have like an inbuilt pattern generator, but it's just they might be responding to cues like olfactory cues, for example, or mental maps. You know, for species that can use mental maps like primates and other species, the knowledge of the environment might also lead to these fractal-like patterns and movement. So that was why I showed this Drosophila larvae with its brain basically deconnected from its spine. Not spine, sorry, they don't have spines, but you get the point. Yeah, you know, vertebrate biologists here. But so even without a brain, without those sensory inputs or you know any kind of memory function happening, they still produce fractal patterns and movements. So that was, and by the way, the people who did that study were heavily criticized. So they're coming back, you know, with different ways of supporting their original claims. I think you had one earlier, yeah.
Okay, thank you. So, if I understood well, you mostly studied animals which were mostly, how to say, relatively free living, like they were not in cages or in like in a territory which is like restricted and stuff like that. So, have there been studies, or maybe you know them, like that they have, for example, such in zoos animals which are really in some restricted territory, that they, I don't know, change slightly some biotic, abiotic changes, and which actually then gave them the opportunity to change their habit again into the normal, like the more complex one.
Right, I do. So not so much my work. I mean, we work with the sheep, so the deworming studies. So those are constrained animals, but still with rather open spaces to use. But these types of studies began with very controlled conditions. So, for example, these toxicology studies, the goal there was, can we use these statistical properties to detect changes in behavior with some kind of introduction of a toxin, right? So with the fish example, it was lead exposure. So they would introduce juvenile fish to a certain amount of lead in the environment, and then measure their behavior as adults. And it was inducing some kind of change. But of course, they can't come back from that. So it's not like you can, in that case, they couldn't, bring the animals back and see if you can restore the regular dynamics. Similarly for mosquitoes, this was done with mosquito larvae in some medicinal plant extracts. So they found that this altered dynamics can be induced by that. There's been studies with rats with the same thing, some pharmaceutical setting. But I've never seen one where you have an animal that's progressed to some different kind of state and then been brought back. So with the polar bear example that I showed, this is kind of something I'm getting more and more interested in is, first, can we use these properties of behavior effectively as bioindicators in captive animals to mitigate those kind of catastrophic changes? But if the animal is already in catastrophe, can we measure it coming back out? So that's maybe the next phase of this research would be quite cool.
Thank you. Are there any methods how to measure or compare similarities between different scales to say, okay, that's the fractal pattern or not? Because I've been looking through those pictures with too much naive eyes, and sometimes it's like, okay, well, I can draw basically any type of curve and say, okay, this is fractal.
I know. Yeah, so that was one of the questions I had when first getting into this research, because I have not actually seen a sequence of behavior that is not fractal. So it's not true. I found it once with great apes in our kind of cognitive experiments when we introduce a computer algorithm that's designed to exploit the chimpanzee's choice patterns. So if they're trying to match or mismatch a computer in a game, they have some inbuilt pattern that each individual falls on, and it usually has some fractal properties to it. But if we gave them a game where the computer is trying to cheat them, not cheat them, but beat them, by using their choice history against them, suddenly I've seen it in two individuals, they just switch to a completely random pattern. So that was quite cool. So it was the first time that I actually got to see a totally random sequence of behavior without fractal properties. And so for me, that was nice because I thought, wow, it's not futile because there are cases where this happened. But the real key, I think, to answer your question is that there's a lot of variation in the patterns we see. Almost never will we see a mathematical fractal emerge from a natural process. So we'll never get to that ideal phase. So when we talk about fractals, what I'm talking about is statistical fractals. And they look
close to perfect fractal.
Yeah. And so you can have a range of some of them may be more ideal than others, but and that means that almost every sequence you see has fractal elements to it, but it doesn't mean every part of the sequence is fractal.
Okay, thank you.
Sure.
At some point of your introduction, you showed that there were some between individuals differences in behavior. So I was wondering if you would think that this method with fractals would be suitable for studies of animal personality.
Oh yeah. I do actually. I dabbled a bit in that. I had some colleagues who study personality, and we looked at whether we could correlate some aspects of an animal's personality with the dynamics of its behavior, but I didn't explore it enough to actually have a intelligent comment on that. But I think probably you have it. So one of the areas I think would be really useful to apply this work is also, it has relevance for personality work. It's like open field testing. So open field testing is something you have a lot in the pharmaceutical world where you're looking at certain responses to some drugs and assessing how it affects this kind of shy-bold continuum, which is a common one in animal behavior studies. Probably you could add this idea to that. I'm not sure what the results would be, but I would expect that the elements that would be most relevant in personality is that tendency to explore versus not, which is very related to the shy-bold continuum. And we can probably suggest that certain fractal patterns in behavior are more or less indicative of exploration. And we've done a study with penguins that looked at that gradient. So there is a lot of variation in kind of exploratory nature of different individuals. But we've never really pursued it to link to something like personality. But it's a good idea.
Maybe another one. Are you aware of any study using the land snails as a land snail?
Yes, actually, there are, and apparently snails also have levy-like movements in them. So, yeah.
I was wondering if there was any researcher having so much time to study because they are moving quite slow.
Very slow, yeah. Yeah, I mean, there's a surprising range of, especially with that levy literature. When that paper was published in Nature in 2007, it kind of started this explosion of studies. Now my species does levy walk, my species does levy walk, my species does levy walk. You know, and then eventually everyone comes in and says, you know, but so what? Yeah.
Thank you also for a fascinating talk. I have just a short question. I think it was on slide number 23, and there were these pictures of parallel relationships, and one of them at the right, bottom right.
Oh yeah.
Completely.
Yeah. Good, thank you. So so this is a good story because when I first read this paper, I thought, okay, great. So every species you measure does a levy walk. It has these patterns of movement. And I thought, wow, it's published in Nature, it must be big news. Yeah, but I failed to see, especially if you agree with all the critics of this approach, where anything can generate levy statistics and movement, I failed to see the value of just demonstrating that species have levy properties in the movement. And this paper was all about just demonstrating that all of these species do the levy walk. That's fine, it's like the first study that had millions and millions of data points. But this is a juvenile basking shark. And in the paper, in the supplement, I think it's even in the supplement, not even in the main paper, they're like, all of the things we tested, except for this one juvenile basking shark, had levy statistics. And then I was like, man, what's going on with the juvenile basking shark? That's more fascinating to me. And so I've written about this, but one of my major criticisms of this is that this whole literature completely ignores individual variation. Right? So even this, it's not like one turtle, it's like all the turtles' behavior combined and averaged to create these general statistics. But there's so much variation across individuals that was completely ignored by these studies. I think now you start to see more and more how, rather than just demonstrating that these statistical properties exist, my whole thing has always been about how can we use that information to understand something about the evolution of the animal and variation across individuals. So I agree with you. That's, to me, the most interesting part of this slide.
Did you also compare juveniles with the other?
No. I think it's just, you know,
it changes throughout the lifetime?
See, I mean, this is a fantastic question, right? Is how much does development influence? So with Japanese macaques, my data show that there is an age gradient. And so juveniles behave in a very different way than adults do. And that, if you think about what levy or what power law statistics mean is, you know, in terms of exploration with the environment, you could easily expect how juveniles should have a very different pattern than adults, just because they're designed to explore at a young age. And then their behavior kind of dampens or becomes more predictable with age. That would be the hypothesis that I would come up with. Yeah. Thank you.
What's the meaning of the data points? The individual or?
No, I think this is I think on this graph, each of them represents an average across all of the individuals that they had from that species. Well, from a study, right? So a study might have, you know, ranging from 5 to 20 individuals. I don't know. Yeah. So that's the average kind of step relationship between the step length to the frequency of it. Sure.
Okay.